Welcome to video series on lipid metabolism, fatty acids and triglycerides, adipose tissue metabolism, fatty liver and lipotropic factors. What you'll learn in this video: synthesis of fatty acids and triglycerides in a simplified way, changes in adipose tissue in well-fed and fasting state, clinical correlation of fatty liver and increased triglycerides, lipotropic factors, important MCQ discussion, probable university examination, and for quick revision. Why is this topic important? From exam point of view, this is a probable question that will be asked. Describe synthesis of formic acid, its energetics regulation. How is chain elongated once a fatty acid is synthesized? Explain how saturated fatty acid gets converted to unsaturated fatty acid. How is triacylglycerol or triglyceride synthesized? What's the difference in triacylglycerol synthesis in liver and adipose tissue? And describe the metabolism of adipose tissue. From patient point of view, this fatty liver, its causes and biochemical basis is very important, and lipotropic factors to prevent development of fatty liver is important. Now let's try to understand the synthesis of fatty acid. This is how the question that will be asked in your university exam. Describe synthesis of formic acid, its energetic and regulation. How is the chain elongated, and how saturated fatty acid can be converted to unsaturated fatty acids. Now, so the starting material for synthesis of fatty acid is acetyl-CoA, and this acetyl-CoA is derived from aerobic glycolysis as end product of aerobic glycolysis or end product of beta oxidation. Now, this acetyl-CoA is formed inside the mitochondria, whereas fatty acid, de novo synthesis of fatty acid takes place in cytoplasm. This acetyl-CoA cannot come out of mitochondria as there is no separate transporter for acetyl-CoA. So, this acetyl-CoA combines with oxaloacetate to form citrate catalyzed by the enzyme citrate synthase. And this citrate has got a transporter in the mitochondrial membrane. So citrate comes out of the mitochondria into the cytoplasm. Now here this undergo lysis with the help of citrate lyase enzyme back to acetyl-CoA and oxaloacetate. Now this oxaloacetate gets reduced to malate with the help of malate dehydrogenase enzyme, because oxaloacetate cannot go back to mitochondria as there is no transporter available for oxaloacetate. So oxaloacetate gets reduced to malate. We have a separate transport protein for malate. Malate enters into the mitochondria. It gets oxidized to oxaloacetate to rerun this cycle. Now, what is the fate of this acetyl-CoA? Now, this acetyl-CoA gets converted to malonyl-CoA by carboxylation reaction. So, acetyl-CoA undergoes carboxylation with the help of carboxylase, acetyl-CoA carboxylase enzyme, and the coenzyme is biotin. So, acetyl-CoA gets converted to malonyl-CoA. Acetyl-CoA, as you all know, it is a 2-carbon compound, and malonyl-CoA is a 3-carbon compound. Now, this fatty acid enzymes, the enzymes that are involved in synthesis of fatty acids are multi-enzyme complex. So they this multi-enzyme complex exists as a dimer. Okay, so to first monomeric unit, activated acetyl-CoA gets attached, and to the second monomeric unit, malonyl-CoA gets attached. It's a very complex one. It is better if you know these steps. That is more than sufficient for undergraduate level. So just only thing you need to remember is this fatty acid synthase complex. It's a multi-enzyme complex with dimeric unit. To the first monomeric unit, acetyl-CoA gets attached to condensing unit, and in the form of acetyl-S-CE. CE refers to condensing enzyme. Whereas this malonyl-CoA gets attached to acyl carrier protein. Okay. So now in the first step, condensing enzyme condenses acetyl-CoA and malonyl-CoA to form beta-keto-acyl ACP. ACP stands for acyl carrier protein, which is carrying the substrates of de novo synthesis of fatty acids. So in the next step, beta-ketoacyl ACP is reduced to beta-hydroxyacyl ACP, and the reduction is done by NADPH. In beta oxidation, it was NADH. Here, the reducing equivalent used is NADPH. Okay, and the enzyme is ketoacyl reductase. So in the next step, this beta-hydroxyacyl ACP undergoes dehydration to form enoyl ACP or alpha-beta unsaturated ACP. In the next step, it gets enoyl ACP gets reduced to butyryl ACP, and the reducing equivalents come from NADPH, and the enzyme is enoyl reductase. Now these three intermediates, or these three enzymes constitutes the reduction unit from beta ketoacyl ACP to beta hydroxyacyl ACP to enoyl ACP and forming butyryl ACP. Now, so with addition of different malonyl-CoA, at the end the palmitic acid is formed, and this has to be released, and the releasing enzyme is thioesterase. So this constitutes the releasing unit. Now this palmitic acid is a 16 carbon fatty acid. Now the chain elongation is done not in the cytoplasm. So the chain elongation can be done either in the mitochondria or in the microsome. So chain elongation in mitochondria is done with the help of addition of acetyl-CoA molecules, and in the microsome is it is done with the help of malonyl-CoA molecules. Okay. For chain elongation, the palmitic acid re-enters into mitochondria or the microsome. So here it gets converted to 18 carbon unit to form stearic acid. Now this stearic acid is a saturated fatty acid. Now how are these unsaturated fatty acids synthesized? They are synthesized with the help of a desaturase enzyme. So we have a double bond at position 9 for oleic acid. Oleic acid is a 18 carbon fatty acid, but it is a monounsaturated fatty acid with a double bond at position 9. So this is added by delta 9 desaturase enzyme. So this is how you need to answer all this question. So describe synthesis of palmitic acid, you have to explain this part. Then how is chain elongated? Chain elongated, either you can write mitochondrial microsome where the chain elongation is done to form stearic acid 18 carbon. Then how unsaturated fatty acid synthesized means by delta 9 desaturase enzyme. This is how de novo synthesis of fatty acid takes place in the body. Coming to regulation and the energetics part. Now this regulation is done by, we have this covalent modification, allosteric regulation, and this feedback inhibition. So feedback inhibition is very easy to remember. So the palmitic acid, that is the end product of de novo synthesis of fatty acid, causes feedback inhibition of acetyl-CoA carboxylase enzyme, which is a key enzyme of de novo synthesis of fatty acids. Okay, so when large amount of palmitic acid is available in our body, it will inhibit the key enzyme, that is acetyl-CoA carboxylase. So there is no more synthesis of fatty acids. Now, so apart from this, the allosteric regulation is done by citrate. So when large amount of citrate is available, it will stimulate this acetyl-CoA carboxylase enzyme. So more and more acetyl-CoA will enter into de novo synthesis of fatty acid. This is about allosteric regulation. Then the other type of regulation which is operating at genetic level is the induction and repression of the enzyme. So insulin is anabolic hormone, and glucagon is catabolic hormone. Now apart from that genetic regulation, these two hormones also bring about covalent modification of acetyl-CoA carboxylase. Now glucagon activates this glucagon phosphorylates all the key enzymes in fasting condition, whereas insulin dephosphorylates all the key enzyme in well-fed state. So what happens is this de novo synthesis of fatty acid is taking place when we have food that is well-fed state, and the boss in well-fed state is insulin. So insulin dephosphorylates acetyl-CoA carboxylase enzyme, and it will make it active, so that de novo synthesis of fatty acid takes place in well-fed state when insulin is the boss. Okay, this is the covalent modification done by insulin. So when we are fasting, what happens is glucagon is active. So glucagon phosphorylates the acetyl-CoA carboxylase enzyme, but this phosphorylated carboxylase is inactive. Okay, so especially in fasting state. So when we are in fasting state, de novo synthesis of fatty acid is not taking place. So this is known as covalent modification of acetyl-CoA carboxylase by phosphorylation of the key enzyme by glucagon and dephosphorylation of the key enzyme by insulin. So what you need to remember is dephosphorylated acetyl-CoA carboxylase is active in well-fed state under the influence of insulin. Okay, so coming to this is just a pictorial representation of the same. So acetyl-CoA is formed either as an end product of beta oxidation or as the end product of aerobic glycolysis. This gets converted to citrate because acetyl-CoA cannot come out of the mitochondria to cytoplasm as there is no transport protein available. So we have a transport protein for citrate. So citrate comes out of the mitochondria to cytoplasm. It is acted upon by lyase enzyme releasing the acetyl-CoA. So this undergoes the different steps to form palmitic acid. the 16 carbon unit. Okay, and what is the fate of this oxaloacetate? Oxaloacetate gets reduced to malate, and we have a malate transport protein, and it will transport the malate back to mitochondria, and here it gets oxidized to oxaloacetate for regeneration of this. Okay, now so the palmitic acid that is formed in the cytoplasm has to undergo for chain elongation. So chain elongation can either take place in mitochondria with the help of acetyl-CoA, or it can take place in microsome, that is endoplasmic reticulum, by addition of malonyl-CoA. So palmitic acid 16 carbon unit gets converted to stearic acid 8 because of the deficiency of the enzyme glycerol kinase. Okay, so this glycerol kinase enzyme is deficient in adipose tissue. That's the reason the TG synthesis in adipose tissue starts from dihydroxyostone phosphate, which comes from glycolytic. Okay, now coming to adipose tissue metabolism. So adipose tissue is of two types, the white adipose tissue and the brown adipose tissue. So this white adipose tissue is characterized by abundant fat with peripherally located nucleus, because this accumulated fat displaces the nucleus to the periphery, and function of white adipose tissue is mainly storage of fat in the form of triglycerides. We have one more type of adipose tissue called as brown adipose tissue, characterized by a large number of mitochondria. Because of the presence of mitochondria, it is known as brown adipose tissue, and it has got the normal nucleus with the large number of mitochondria. So the main function of this brown adipose tissue is thermogenesis. This is the electron transport chain, which has got 5 different complexes: complex 1, 2, 3, 4 and 5. Now hydrogen ions are pumped out of complex 1, complex 3 and complex 5, and this hydrogen ion enter through complex 5, and this causes phosphorylation of ADP to ATP. So what this brown adipose tissue does is, this has got a uncoupling protein called thermogenin, which causes which inhibits the entry of this hydrogen ion through complex 5, and it will inhibit conversion phosphorylation of ADP to ATP. So this is dissipated as heat. So thermogenesis is the important function of brown adipose tissue. White adipose tissue is concerned with storage of triglycerides, and brown adipose tissue is concerned with thermogenesis. So the question that can be asked in your exam is, describe adipose tissue metabolism. So you have to describe the metabolism both in well-fed state and in fasting or the starvation state. So when we have food, what happens? The lipids are broken down to what? Triglycerides, cholesterol esters and phospholipids. Now check out for the digestion absorption of lipids the first video of lipid metabolism. Now this long chain fatty acids are esterified with the glycerol to form triglycerides, and the free cholesterol is also esterified to cholesterol esters, and phospholipids all are packed into chylomicrons with ApoB48 and A as apolipoproteins, and they are released into general circulation through lymphatics. Okay, simultaneously what happens? Liver is the acetyl lipogenesis is also taking place in liver. So the acetyl-CoA, the starting material for denosynthesis of fatty acid through multi-enzyme complex, gets converted to palmitic acid or fatty acid. This is esterified with glycerol to form triglycerides. Along with triglycerides, now these triglycerides are packed along with triglycerides, cholesterol esters, they are packed into very low density lipoprotein, and this is the endogenous triglycerides, which are packed into very low density lipoprotein, and this VLDL has got ApoE and ApoB100 as apolipoproteins, which are carrier proteins. So chylomicrons carries the exogenous triglycerides, and VLDL carries the endogenous triglycerides. Now this VLDL is also released into circulation. Now this C2, which is present in VLDL, and C2 which is present in chylomicron, and this C2 is donated by HDL2 to chylomicron. Okay, so initially it will have only ApoB48 and A. So the C2 and E are donated by HDL2 or to chylomicron. Okay, now this ApoC2 will activate lipoprotein lipase that is present in the endothelial cells of the blood vessel, and this will will hydrolyze the triglyceride to glycerol and fatty acid. Now this glycerol is taken up by the hepatocytes and the fatty acids are taken up by adipose tissue. Now, so in the adipose tissue, again it gets stored as triglycerides. Now, as we all know, there is glycerol kinase is deficient in adipose tissue. So the glycerol part is comes from dihydroxyacetone phosphate from glycolysis. Okay, so this is how this dihydroxyacetone phosphate to this dihydroxyacetone phosphate fatty acids are attached and it forms triglycerides and gets stored. Okay, so this is about and this storage of triglycerides in adipose tissue is under the influence of insulin. So this insulin stimulates both glycolysis and lipogenesis. Insulin is an anabolic hormone which is active in well-fed state, and this will stimulate the triglyceride synthesis and storage in adipose tissue. This is about metabolism of adipose tissue in well state. So now how this adipose tissue changes its metabolism whenever we are fasting or starving. Now in fasting or starvation, this condition is under the influence of hormone glucagon. Okay. Now this glucagon will stimulate adenylate cyclase enzyme which causes cyclization of ATP to cyclic AMP. Now this cyclic AMP in turn converts or activates inactive protein kinases to active protein kinase. Now this active protein kinase will cause phosphorylation of the enzyme hormone sensitive lipase. Now this phosphorylated hormone sensitive lipase will break down the triglycerides that are stored in adipose tissue to fatty acids and glycerol. Now this glycerol is taken up by the liver and gets converted to dihydroxyacetone phosphate which will enter into glycolytic pathway, and these fatty acids are taken up by the muscular system, skeletal muscle, smooth muscle and the cardiac muscle, mainly by the skeletal muscle where they undergo beta oxidation to provide energy. The end product of beta oxidation is acetyl-CoA. Now this acetyl-CoA will enter into TCA cycle and provides energy when we are not having any food. So this is about metabolism of adipose tissue in condition. Now this adipose tissue secretes adipokines. It is just like so the adipocytokines are also called as adipokines or adipocytokines. There are three in number: leptin, resistin and adiponectin. Now they have got different roles in our body. Leptin, whenever we consume large amount of fat or when large amount of triglycerides are present in the adipose tissue, it will stimulate the synthesis of leptin. So this leptin will in turn stimulate neuropeptide Y of hypothalamus, and this will decrease the appetite. So this is one side. Leptin is considered to be a regulator of satiety or hunger. Okay, so whenever there occurs mutation in this leptin biomolecule, or when the concentration of leptin is decreased, it results in the development of obesity, increased BMI. So one more hormone is there called adiponectin. Now this adiponectin is known to enhance the insulin sensitivity. Okay, so decreased adiponectin is also related to development of obesity. So decreased leptin, decreased adiponectin are related to obesity. So we have one more adipocytokine released by the adipose tissue called resistin. Now this resistin is known to stimulate inflammation, okay, by releasing CRP, C-reactive protein, interleukin 6 and tumor necrosis factor alpha. So increased resistin is dangerous to the body because it will stimulate chronic low grade inflammation, and that also has been related to obesity. So sometimes a 3-marker question is asked like enumerate different adipocytokines released from adipose tissue, their functions and clinical significance. This is the simplest way to remember. So leptin, regulator of appetite; resistin stimulates inflammation; adiponectin is stimulates insulin sensitivity. So decreased leptin, decreased adiponectin are related to obesity, and increased resistin is also related to obesity. Now coming to what is the link between adipose tissue and obesity. So So whenever we consume carbohydrate rich diet, you all know it is ultimately broken down to glucose, and this glucose stimulates the insulin release by beta cells of pancreas, and adipose tissue has got insulin receptors because it is dependent on insulin. So this insulin binds to the insulin receptors on the adipose site, and this causes translocation of the GLUT4 from the cytoplasm to the cell membrane of the adipocyte, and this is how the glucose enters. Now this is a normal adipocyte. So when we consume large amount of calories, what happens, there occurs hypertrophy. So hypertrophy means what? Increased in the size of the adipocyte, and hyperplasia also, increase in the number of the adipocyte. So this will alter the insulin receptor, and insulin can no longer bind to the adipocyte. So when insulin does not bind to adipocyte, what happens? The GLUT4, which is present in the cytoplasm, cannot be translocated to the cell membrane, so glucose cannot enter, and this will result in insulin resistance. Okay, so this insulin resistance and excess and with insulin resistance, and what happens, and when a person continues to eat, so there is hypertrophy and hyperplasia of the adipocyte, and the TG is no longer inside the adipocyte, it becomes extracellular, and this is not metabolically active, and it constitutes dead bulk. Okay, and this increases the this results in the development of obesity. So how to measure obesity? Using body mass index. Now we can measure this body mass index by taking into consideration weight and height of the individual. So weight by height square will give us our body mass index. If the body mass index is between 18.5 and 24.9 kg per meter square, then the individual is considered to be a healthy individual or a normal individual. If the BMI lies between 25 and 29.9 kg per meter square, the person is overweight. If the BMI is more than 30 kg per meter square, then the obesity terminology is used. This obesity is of two types, central and the peripheral. So central obesity is also known as visceral or the android obesity, characterized by fat accumulation around the abdomen, and this is associated with increased risk of all metabolic diseases. Whereas the peripheral obesity or the gynoid type of obesity is characterized by fat accumulation in hips and thighs, and this the metabolic disease risk is relatively less when compared to central obesity. Now coming to what are the complications of obesity because of altered adipose tissue metabolism. Check out this. So whenever we consume excess calories, what happens is the excess carbohydrate is getting broken down to glucose. This will stimulate the release of insulin, and insulin will go and bind to the insulin receptor that are present on the adipose tissue cells, and this causes translocation of the GLUT4 from the cytoplasm to the cell membrane so that glucose enters. So when we are consuming excess calories, what happens? There is altered insulin receptor. So excess calories causes what? Hyperplasia and hypertrophy of the adipose tissue cells, and the TG becomes extracellular, and this acts as a dead bulk because it is not metabolically active. Now this alters the insulin receptors, so insulin resistance. Insulin is there in the body, but it cannot act on the adipose tissue. This results in insulin resistance. Because the glucose is not able to enter, so it results in hyperglycemia, and chronic hyperglycemia results in the development of type 2 diabetes mellitus. So because there is insulin resistance and the glucose is not being taken up inside the adipose sites, so there occurs lipolysis. Because the glucagon ratio increases, and this will stimulate lipolysis, there occurs increased free fatty acids released from the adipose tissue cells. Okay, now so this excess calories which causes hypertrophy and hyperplasia of the adipocytes increases the BMI. So, and because of increased free fatty acid, there occurs a state of dyslipidemia, altered lipids. Now so this can lead to development of non-alcoholic fatty liver disease. Now this can result in accumulation of fat in the liver, predisposing to the development of non-alcoholic fatty liver disease. What was the previous assumption is fatty liver was mainly seen among alcoholics. Now here the person is not consuming alcoholic, but still develops fatty liver because of insulin resistance. Okay, now this dyslipidemia can cause endothelial dysfunction, and this can decrease the nitric oxide. So check out for the functions of the nitric oxide in videos on protein metabolism. Okay, so decreased nitric oxide, what happens? So the nitric oxide main function is vasodilatation. So decreased nitric oxide means decreased dilatation of the blood vessels. As a result of that, BP increases, predisposing the individual to the development of hypertension. Apart from that, dyslipidemia is also known to stimulate renin, angiotensin, aldosterone system. So increased angiotensin 2, again it's a vasoconstrictor, so the blood pressure, systolic blood pressure increases. So increased aldosterone, which causes retention of sodium and water, so this also increases the blood pressure, predisposing the individual to the development of hypertension. Apart from that, dyslipidemia, accumulation of the lipids in the cardiac blood vessels, medium and the large size blood vessels, results in the development of coronary artery disease, atherosclerosis, and can predispose the development to myocardial infarction. Apart from that, this dyslipidemia, the lipids can also get accumulated in the pharyngeal walls and the neck, and this can narrow the upper airway. So partial or complete airway obstruction during sleep results in interrupted breathing in the form of apnea. So also predisposes to the development of obstructive sleep apnea syndrome. Apart from that, this hypertrophy and hypoplasia of the adipose tissue, so this will increase the load on the joints and also stimulates chronic low grade inflammation, predisposing the individual to the development of osteoarthritis. Now just check out, simple excess calorie intake is associated with so many complications. Just to summarize this complication, it can predispose to the development of type 2 diabetes mellitus, non-alcoholic fatty liver disease, hypertension, atherosclerosis, subsequently myocardial infarction, obstructive sleep apnea syndrome, and arthritis. Coming to the last topic of this video, fatty liver and factors. Now what are the causes for fatty liver? So the first important cause is excess calorie intake in the form of excess consumption of carbohydrate or excess consumption of lipids. So excess carbohydrate, so the end product of anaerobic glycolysis is pyruvate, and pyruvate gets converted to acetyl CoA. This acetyl CoA will predispose to the development of what denosynthesis of triglycerides, and that is how the fats increases and fatty liver can occur. Okay, so the second important cause for fatty liver is diabetes, uncontrolled diabetes mellitus, and starvation also causes fatty liver. This is because so in diabetes mellitus and starvation, the insulin glucagon ratio is reversed. There is more of glucagon which will stimulate lipolysis, because so this glucagon will stimulate hormone sensitive lipase and will cause breakdown of triglycerides to fatty acids and glycerol. These fatty acids again go back to liver and they get converted to endogenous triglycerides, and that is how the fatty liver occurs. The third one is the second cause is mobilization of fat from the adipose tissue due to uncontrolled diabetes mellitus and starvation. So this is the reason, biochemical basis. The third one is toxic injury to the hepatocytes in the form of drugs and poisons, and this can also impair the clearance of the fat from the liver as VLDL, and so what happens, the triglycerides accumulate in the triglycerides or the fatty acid get accumulated in the liver, resulting in fatty liver. So the next important cause is alcohol, chronic alcoholism. What happens, alcohol is getting oxidized to acetaldehyde and NAD is getting reduced to NADH. Now this reduced NADH causes reduction of oxaloacetate to malate. So what happens is oxaloacetate is not available for operation of TCA cycle. Now this acetyl CoA gets diverted to form fatty acids and they accumulate in the liver of the alcoholics, predisposing to the development of fatty liver. Okay, so the one more condition is non-alcoholic fatty liver disease. Here fat accumulates in the liver in absence of consumption of alcohol, mainly because of insulin resistance, which has been explained in the previous slides. So what is the ultimate outcome of this fatty liver is, so accumulation of the fat in liver is known as steatosis. So this will stimulate low grade inflammation called steatohepatitis. Now if left unchecked, this leads to development of fibrosis where there occurs proliferation of the collagen. If left unchecked, there occurs diffuse collagen formation in the form of cirrhosis, and this can result in malignant transformation of the hepatocytes to hepatocellular carcinoma. The first two stages where there occurs accumulation of the fat and low grade inflammation, these two stages are reversible. Subsequently, once the fibrosis sets in, which will progress to cirrhosis and hepatocellular carcinoma, and this becomes irreversible damage to the hepatocytes. Now all these can be prevented when lipotropic factors are consumed. Now what are lipotropic factors? Now these are the substances that help prevent fat accumulation in the liver by promoting transportation and metabolism of lipids. How do these lipotropic factors act? They promote synthesis of phospholipids, especially phosphatidylcholine, that is lecithin. They help in formation of very low density lipoproteins, wherein they pack the fatty acids esterified with glycerol and convert this into triglyceride. They pack them into very low density lipoproteins and they are released into circulation. That is how they export the triglycerides from the liver and they release into general circulation. Now, which are these lipotropic factors? And these include choline. Now, choline combines with choline forms phosphatidylcholine and which is lecithin. So, this fat gets converted to phosphatidylcholine. That is how it prevents accumulation of fat. Next is methionine. It donates a methyl group and it helps in formation of choline synthesis. So inositol, it is also a component of phospholipid, helps in transportation of lipids and prevents its accumulation. Folic acid and vitamin B12 helps in conversion of homocysteine to methionine. That is how they act as lipotropic factors. So this was about fatty liver and lipotropic factors. Fatty liver can be asked as a case-based question, and lipotropic factors can be asked either as a 5-marker question or a 3 question. So this was all about the synthesis of fatty acids, triglycerides, adipose tissue metabolism, and fatty liver and lipotropic factors. Coming to the assessment part, rate limiting enzyme of fatty acid synthesis is acetyl-CoA carboxylase. The cofactor required for acetyl-CoA carboxylase for all the carboxylation reaction, it is pentose phosphate pathway or the HMP. Okay, so fatty acid synthesis occurs mainly in cytoplasm. Beta oxidation occurs in mitochondria, and fatty acid synthesis occurs in plasm. Okay, so fatty acid synthesis occurs in cytoplasm. Repeat question. The immediate product of fatty acid synthase complex is what? Palmitate is the major fatty acid that is synthesized by de novo synthesis. Okay. Transport of acetyl-CoA from mitochondria to cytosol occurs by citrate. As there is no transport protein for acetyl-CoA, so it utilizes citrate cycle to come out from the mitochondria into cytoplasm. So malonyl-CoA inhibits carnitine acyl transferase 1. So this CAT1 is the enzyme associated with the transfer of fatty acids from cytoplasm to mitochondria for beta oxidation. So malonyl-CoA formed will inhibit carnitine acyl transferase 1 enzyme, so that mainly to prevent butyl cycle. So when fatty acid synthesis is taking place, beta oxidation is inhibited. Now, so number of acetyl-CoA molecules needed to form palmitate is 8 acetyl-CoA to form 1 molecule of hormone that stimulates fatty acid synthesis is insulin. Insulin is an anabolic hormone, so it stimulates genesis. Fatty acid synthesis uses which reducing equivalent? Mainly utilizes NADPH, whereas beta oxidation utilizes NADH. Yeah, so backbone for triglyceride synthesis is glycerol 3 form. Okay, major site of triglyceride synthesis, it takes place both in liver and tissue. Enzyme absent in adipose tissue for TG synthesis is glycerol kinase. It is mainly present in liver. In adipose tissue, it utilizes dihydroxyestrogen phosphate obtained from glycolysis. Source of glycerol 3 phosphate in adipose tissue is glucose from glycolysis, which forms dihydroxyestrogen. Okay, fatty livers occurs due to impaired synthesis of VLDL. Okay, so VLDL, very low density lipoproteins. The fatty acid that is synthesized in the liver is esterified to glycerol to form triglycerides, and this is released into circulation, which carries the endogenous triglycerides. Which amino acids acts as lipotrophic factor? It's mainly methionine. It provides methyl group and helps in formation of choline synthesis. Deficiency of which vitamin can lead to fatty liver? It is B12, because we know B12 is required for conversion of homocysteine to methionine. So indirectly it affects lipid. Which conditions commonly associate fatty liver? So starvation, lipolysis, because glucagon ratio is high, obesity, increased accumulation of triglycerides, alcohol also. What happens? There is increased NADH, so decreased oxaloacetate, no TCA cycle, so acetyl-CoA gets diverted to form denosynthesis of fatty acids. So in all these conditions, fatty liver is. So alcohol-induced fatty liver is mainly because of increased NADH. So increased NADH, fatty acid oxidation decreases and it stimulates fat. So just check out your score on this topic. If the score is more than 50%, it's good, highly appreciable. Less than 50%, do redo. Please do rewatch this video and retake all the MCQs. So this was all about lipogenesis. Next video will be on cholesterol metabolism. So please do like, share and subscribe. Thank you. Happy learning.